Hello, folks. Welcome to day 2 of trimester 2. Today, we're going to start diving into the function of neurons and talking about how those types of cells perform their specialized function. When we talked yesterday in our brief introduction to neurons, we talked about the fact that they use a couple different types of signaling. They use electrical and chemical signaling. And so as signals travel along a neuron, like along one cell, that is electrical signaling. But then when it needs to hop from one neuron to the next, that's chemical signaling. So an electrical signal and that chemical signal then triggers another electrical signal, which triggers another chemical signal that goes from here to the next neuron. So it's kind of this alternating pathway of electrical and chemical signaling. And as you probably guessed from that vocab up on the screen right now, today we're going to be focused on the electrical signaling that happens along the axon. That's this long, skinny part of a neuron. So let's dive into this here. As we start talking about this, one of the things that's really important is the movement of ions in and out of the neuron. And so that happens through transport across the cell membrane. And we talked about passive transport in the last unit, but today we're going to talk a little bit about active transport. As we talk about this, and as we talk about some other topics that are going to be coming up throughout this unit, one really important thing is to remember how protein structure is determined. Because as you can see from this picture up at the top, this is an image that shows all of the different proteins that play a role in the signaling that happens in neurons. There are a lot, and their shape is super critical. So if you remember, the primary structure is that sequence of amino acids. And the secondary structure is caused by hydrogen bonding between the backbone, but what really determines the unique three-dimensional shape, that tertiary structure, is the interaction between the R groups of those amino acids, those unique different pieces that can form hydrogen bonds or ionic bonds or covalent bonds that all pull that molecule together into a separate shape. And so if there's anything that interferes with those bonds, or affects how those bonds form, that's going to change the shape of the protein. That's going to be really important just to keep in the back of your mind as we go through the next few topics in this class. So today we're talking about active transport, and it's called active transport because it requires energy and it moves against the concentration gradient. So this is different from passive transport that was like rolling down a hill. Active is like walking up a hill. It takes energy and you're going against the gradient, so from lower to higher concentration. This is accomplished by things called carrier proteins that can hold on to molecules and change shape to push those molecules across the membrane. And it's that shape change that really requires energy here. This allows the inside and outside of the membrane to have different chemistry. And you'll see with neurons why this is so important in just a minute. One of the important carrier proteins that functions a lot in the nervous system is called the sodium-potassium pump. And we're going to watch here for a minute how that works. As you watch this animation, there's no sound on this. But just pay attention to what's going in and what's going out. So you can see this protein has a really unique shape in the membrane. This is the inside of the cell. Sodium ions fit into particular places in one part of the cell. Here, ATP bonded down here and released a phosphate, and that released energy that caused this protein to change shape and release those sodiums. Remember, phosphate groups are negatively charged, so that negative charge is going to interact with the R groups of all of those different amino acids to change the shape of that protein. And when that phosphate is released, it changes back and brings two potassium ions into the cell. So every time this pump functions, you have 3 sodiums that go out and 2 potassiums that come back in. And both of those ions are moving against their concentration gradient with the help of energy from that ATP molecule. So this pump, this sodium-potassium pump, helps to maintain a difference in ion concentration. And because those ions have charge, this is something we call membrane potential. Voltage, remember, is a difference in electrical charge. And electrical charge is those positives and negatives. And so if you have more positive charge on one side than on the other, you have created voltage, which is also referred to in cells as membrane potential. This is important in neurons because it acts as a source of energy for the movement of any charged substances. Think of this like potential energy, just like a battery stores energy in the form of voltage. You can attach it to a circuit and it causes electricity to flow. It also causes things to move across a membrane. And so we're going to see this functioning in several different places throughout this trimester, but this is important in how neurons send those electrical signals down the axon as well. So you're going to go and do a quick modeling activity here on a Jamboard to look at how the membrane potential is created along the axon of a neuron. So as you saw in that activity, both passive and active transport have a role in this. Active transport functions through that sodium-potassium pump, which you can see diagrammed on the right here. Passive transport is those potassium leak channels that are a form of facilitated diffusion that just allow those potassium ions to continuously kind of slowly diffuse out. The combination of these two maintains that voltage across the membrane. The inside of a neuron is negative compared to the outside. And so because of that, cations tend to move in one direction and anions tend to move the other. Remember, cations are positively charged, so they tend to move into a cell, and negatively charged particles tend to move out according to their electrical gradient. But remember, ions move both due to their chemical gradient, that's the concentration, and their electrical gradient. So some of them, like those potassium ions you were just playing with in that simulation or that modeling activity, the concentration gradient actually causes them to diffuse out of the cell, but the electrical gradient pulls them back in because they're positively charged, so they're attracted to the negative interior. So those two forces kind of balance themselves out and help keep those potassium ions kind of at an equilibrium point. The sodium ions are moved out of the cell by both of their gradients. The concentration and the electrical gradient move them out of the cell. And so that's why we require that sodium-potassium pump to help move those. Ions will diffuse down their gradient. That's a combination of both of these, their electrochemical gradient. So these types of pumps, like the sodium-potassium pump, are called electrogenic pumps because they create that electrical difference. This is the main electrogenic pump that functions in animals, the sodium and potassium pump. There are others. There's a proton pump that is very commonly found in plants and fungi and bacteria. And it's just what it sounds like. It pumps protons out of the cell, but it has the same kind of effect. It creates a more positively charged environment outside the cell and keeps the inside of the cell more negative. So you still have that stored energy, that potential across the membrane. That energy, that stored energy, can be used for work. One example of that is ATP synthesis. Making ATP uses energy from this gradient, so that'll come back again there. It's also used, however, to help with some other types of transport. Co-transport is kind of a combination between active and passive transport. Some cells so this process, an analogy you can think of here is we use energy to pump water up a hill, but then as the water comes back down a hill, we could make that water turn a wheel to power something, right? So we have to put energy in to get the water up, but then as it comes back down, we can use that water to do work. The same thing happens in cells. They pump those hydrogen ions out using ATP. So this part is active transport. But then passive diffusion happens because those hydrogen ions come back down their concentration gradient. And as they do that, this protein that allows them to diffuse back in also grabs some sucrose, in this case, a sugar, and transports that back in using the energy that's released because of this diffusion. So co-transport uses that electrochemical gradient as a source of energy to pull in other substances that the cell needs as well. There are other types of active transport besides these carrier proteins, these protein pumps. The sodium-potassium pump is the one that's the most important in terms of the nervous system. But there are some other types that you probably remember hearing about. And these are ways that cells take in or get rid of larger molecules like proteins that a cell makes or polysaccharides or things like that. These substances cross the membrane packaged into vesicles, so little bubbles of membrane, and it takes energy to reform the membrane into those little bubbles. Exocytosis, like think exiting the cell, is the process of moving things out. Endo is bringing things in. And these images kind of show you what how that looks. So here's a vesicle. Maybe these green dots are proteins that were made in the cell. That vesicle comes and fuses with the membrane, and as it becomes part of the membrane, it releases its proteins. Endocytosis forms a vesicle around substances that it wants to pull in. They look like opposite processes. The detailed mechanics of how this happens are different. And both of them require energy because they're rearranging all of those lipids so substantially. Endocytosis, that process of bringing things into the cell, also has a couple different types. If the cell's taking in a larger particle, we call that phagocytosis. Those words literally mean cellular eating. So it takes in a large particle. We call that a food vacuole. Penocytosis, that literally means cellular drinking. It's when a cell takes in little droplets of fluid that contain molecules that are dissolved in it. So taking those little droplets in is a way to get all of the molecules that are out there. If a cell wants one particular molecule more than others, then it will use something called receptor-mediated endocytosis. So here, just let me get this up here so that the picture's up here. Here you can see the cell membrane has specific receptor proteins that are all kind of clustered within an area of the membrane. And those receptor proteins in this case are purposely grabbing hold of those star-shaped molecules. And I'll move myself out of the way here. So as it grabs a bunch of those, it forms this vesicle, and it's possible this vesicle could still trap a little blue square or something here, but they get a much higher percentage of the desired molecule, in this picture, those little stars, than they would without those receptor proteins. So this is how your cells take in molecules that they need specifically, like cholesterol, that they use for the cell membranes and for making steroids. So those are some different types of active transport and a little bit about how active transport and passive transport function in determining the resting potential, that membrane potential of a neuron. And neurons need to have that difference in voltage so that they can send their electrical signals. And we'll talk a little more about how those signals actually transfer down the axon on Monday. Take a look in classroom and see if there are other things that you need to do today. Otherwise, have a great rest of your day and have a great weekend.